Okay, why don't we get going here? It is my pleasure this afternoon to introduce Rava Azeredo da Silveira from direct to us from Paris. You may or may not know he is the CV Star visiting faculty fellow for the semester. So he's been around for a little while. Rava and I are co-teaching a new graduate course on microcircuits in the Retina. And he will continue to be here through January and possibly a little bit into February. Who knows? Many of you know him, but for those of you who don't, he's going to give a little bit of kind of a set of different vignettes of some different research topics he's been engaged in, and so that might kind of trigger interest in various of you out there. So feel free to kind of contact him afterwards, because he's going to be around for the rest of the semester. So Rava starts from a physics background. He did a PhD at MIT in statistical physics. Then he was a junior fellow at Harvard for several years. And after that, he started as a faculty at École Normale Supérieure in Paris, where he still is. And he is Rava is a kind of theorist of the old school type who actually solves hard equations with pencil and paper some of the time. You'll kind of see that come through in some of his work. He's got a number of interests and has engaged in collaborations with a number of different neuroscientists, and he's going to try to give you a little bit of a sampling of a few of those different research projects. So let us welcome Rava.
So thank you very much. It's I'll say just one word before starting, that it's very nice to be here, and one reason for which it's very nice for me to be here is that Roberto Carr is here. I don't know if many of you in neuroscience know him. Professor Carr is a faculty here in more departments than I can remember. And the reason I'm very grateful is that I took my first undergraduate quantum mechanics course with him, which was by far the most exciting physics undergraduate course. Then I did a micro research project with him, but that was my first taste of reading papers and research. And then it was, to a large extent, under his influence, that I went to the US to do graduate studies. So I think if it weren't for him, I would have followed probably a different path, and I wouldn't be here. So, as Michael mentioned, he suggested that I give kind of an overview of a set of different problems. And generally, I'm reluctant to adopt this format, because I find it compresses the space between incomprehensible and trivial to almost zero. And I decided I would go along with the suggestion, because I think the aim of this lecture is much more to give you a little bit of a flavor of different things I've been thinking about, and to explain any specific result or methods or derivation. And so in this thrust, I'll focus really a lot on conceptual points. And so I hope you will forgive me for completely overlooking explaining context or background or derivations, and I hope you'll forgive me some of the conceptual points seem not so original to you. Now, when I was preparing this lecture, I was thinking, could I find some unifying thread along which I can organize it? And I decided that it's not possible. And the reason it's not possible is kind of maybe a little bit interesting in itself, and at least to me it's a mystery, and it's the following: that we have kind of a nesting of computations in neuroscience. There are very sophisticated computations that occur at all scales, and moreover, the computations kind of repeat. So at all scales there is adaptation, at all scales there is feature detection, and to me at least it's a big mystery of how is this nesting organized. And unless one understands or has some rough idea of how it's organized, I don't think really one can give a unified view. And so this is why I called this talk 6 Neurons in Search of a Model. Many of you have guessed this is in reference to a play by the Italian writer Luigi Pirandello, and the play goes like this. There's a director and a set of actors, and they're rehearsing for a play by Pirandello. And in come 6 characters. And these 6 characters are from another play, only they're lost, and they're looking for a director to finish their play. And so the actors will play the role of the characters, and the characters will correct the actors. And so there's this very confusing nesting that leaves the spectators in a general state of confusion at the end of the play. A little bit like the general state of confusion in neuroscience. So I will start, and maybe the first half of the lecture will be discussing one example of computations at the level of circuits, and then I'll go rapidly over some more examples. So what do I mean by a circuit? Well, yes, there's another reason why I refer to this play. Maybe I'll tell you later on. So a circuit, in my mind, is a small number of neurons, a relatively small number of neurons, but certainly a small number of cell types, and of which we know the circuitry, and we know in some detail the physiology of these cell types, and so we're able to ask questions of detailed mechanisms and to view this as a small computational device. Now, I'm going to illustrate this kind of setting with one circuit, which is a circuit that we found in the retina of mouse, and which responds selectively to objects that are approaching the animal. Now, one thing that I've absorbed from discussions with John Hopfield actually here is that a good way to think, a good mental guide to think about computations in the early sensory systems is to have in mind what one can do effortlessly and what is behaviorally relevant. And so you have no problem telling this, or identifying this organism, whether it's in a bright light or in dim light. And also, it appears that the analysis of motion is very behaviorally relevant. So you have no trouble telling whether this organism is flying to the left, to the right, whether it's running away from you or running towards you. Now, this is maybe a little bit of an unusual stimulus, and you may think that it's not very relevant. But if you were a mouse and what was running towards you is an eagle, of course it would feel much more relevant. Now if you think about an eagle, when it flies from very far in the sky to a few meters away from the mouse, it will go from covering about a fraction of an angle on the retina of the mouse to about 10 degrees on the retina of the mouse. And that's the right scale for cells in the retina to detect it. Okay, so how did we come to look at this circuit? So this is how
when you say the right scale,
what I mean by this is that
range of scale.
Right. So what I mean by this is that the receptive fields of ganglion cells in the retina are on this order, and actually the receptive field of the ganglion cell we're going to look at is exactly of this order.
On the 10 degree order.
Yeah.
Okay, so it can't tell the difference between the moon and the eagle far away.
Well, presumably it can, at least if it's looking at it statically with this ganglion cell.
Yeah.
Okay. Well, okay. So, the way it happened is that a collaborator of mine, a friend of mine called Boton Roska, who actually did in his lab the experimental part of this work, came to visit me in Paris and told me, well, we've discovered a very particular circuit in the retina, and we wonder what it does. And so I'm sure most of you are familiar with the general circuitry of the retina, but for those who aren't, you can think of the retina as having 3 layers. There's an entry layer which is made up of the photoreceptors, which convert light into neural signals. There's an output layer here made up of these cells called the ganglion cells, which project to the brain. And these are connected, the 2 layers are connected by this intermediary layer made up of so-called bipolar cells. All these cells are excitatory. And now intermeshed in this network, there are also inhibitory cells like this one, which is called the A2 amacrine cell, and which would concern us. So what he told me was this. He said, we found this ganglion cell, which is kind of original because it receives inputs from 2 pathways. It receives input from an excitatory pathway, and this pathway is off. So this ganglion cell will be excited by dark. Okay? And then it receives inhibition from this pathway, and this pathway is on. So this ganglion cell will be actually inhibited by light. The other peculiarity of this system is this object here, which is an electric synapse, a gap junction between this bipolar cell and this amacrine cell. And the role it fulfills is that it allows the inhibition to arrive very fast. Inhibition arrives more or less as fast as excitation, which is kind of unusual. So this circuit as a whole was unusual, and the question that was posed to me is why. So I thought a moment about it, and I figured, well, if I forget about the biological details, I can think of this cell as follows. I can think of this cell as getting input from many, if you want, yin-yang subunits. So I have excitatory off subunits and inhibitory on subunits. And so I figured, well, if you have a stimulus that has a component which is on and a component which is off in it, then they should cancel, and the cell should not be able to respond. So what kind of stimulus is that? Well, it's clear it's a stimulus that moves laterally. Because if I compare two different times, what I see is that the leading edge here is actually generating a dark crescent, and the trailing edge is liberating some brightness. And so if this dark thing excites the ganglion cell, this bright object inhibits it, then it shouldn't respond. So Botton told me, tomorrow we'll do this experiment. And I didn't believe him, but at the time I didn't know him well. And so 2 days later he called me and said, we did this experiment, works perfectly. He was very excited. So I got very enthusiastic. I went into the shower. It's the best place to think. And I figured, so what can excite this cell? If this doesn't excite the cell, I figured it's a stimulus that is only off. And it's obvious what is a stimulus that is only off is a stimulus that is approaching you. So if you have a dark object that is approaching you, it's generating some annulus of dark in time, and so that you only have an off stimulation of this cell, so none of the inhibition is activated and it should respond.
Rava, just a quick question. Presumably the cells, the yin-yang cells that you described, don't interact with one another.
Yeah, to first approximation, let's think about it this way. We haven't looked at it. I'm sure they do. We haven't looked at it. Okay, so this is a cartoon. This is a real experiment. So the way the experiment is done, you flash, suddenly a bar, let's say, appears on the gray background, and the cell responds to the appearance of the bar, and then at some point you start moving. And either you expand the bar here, that's mimicking an approach motion, and you see a big response of the cell, or you move the bar laterally and the cell barely responds. Okay? Now this is for one velocity, but you can repeat this for a number of velocities, as we did here. And we find that even for relatively slow velocity, of course, the response becomes more shallow, but you have a robust response. And in fact, the number of spikes is relatively conserved. So it's very robust as a function of velocity. By contrast, if you do the same experiment with a laterally moving object, you see no response, essentially no response at any velocity. And it's not that your cell is dead, because you see the cell responds when you're flashing the object, when the object first appears. Okay. We tried also more confounding stimuli, which are stimuli in which the two edges can move independently. And as we expected, there's only a response of the cell in the quadrant where at least one of the edges is moving outward, so where you can maybe interpret the motion as an approaching motion. Okay? So now we had kind of narrowed down the phenomenology, we had a relatively good idea of the circuit, and so making a model, it was very easy. We just had to kind of put these elements together. So we have some stimulus, we have these yin-yang subunits, they arrive at the ganglion cell and they're somehow summed, and then we have to calculate some output. Okay? Of course, one has to take care of the spatial details of the model, and the spatial details go as follow. The receptive field, so that's coming also back to your question, the receptive fields of the yin-yang subunits of this bipolar cell and the amacrine cells are very small. That's another peculiarity of this system, is that it uses a very small amacrine cell comparatively. And there are about 20 of them in linearly across the receptive field of the ganglion cell. So we put those elements together and just wrote down a simple model. So in this model, each of the yin-yang subunits are described as linear nonlinear model, as two linear nonlinear models. So these are very standard model which consist in convolving the input with some linear filter then taking the resulting quantity, passing it through some static nonlinearity. And this yielded some excitatory conductance. The analogous calculation for the inhibitory subunits yielded inhibitory conductance. And then these were summed, and a simple, the potential of the ganglion cell was calculated with a simple, say, RC circuit. And we just had some ad hoc linear threshold linear nonlinearity to predict the spiking rate, because of course it has to be positive. So what's kind of nice with this model is that there are barely any free parameters. Okay, this is measured. These filters, these nonlinearities are measured. The only really free parameter is this very ad hoc thing, but that's just to get some positive number, some spiking rate. Okay, so we were quite happy that this works very well. So here I didn't tell you before, in the figure you saw before, the red traces are data and the dotted lines are the model. And as you can see, they agree fairly well for such a simple model, and here also they agree quite well. Now, I'm a little bit surprised that none of you have come up with a criticism, like the sharp minds, the sharp critical people from Bell Labs haven't come up with a criticism of this.
So why did you have what's the role of the fast junction?
Okay, so the role of the fast junction is that the inhibition arrives early enough to cancel the excitation. Otherwise, you would have a little moment where you have just excitation and the ganglion cell would respond. Okay. So, yes?
I'm thinking about moving along a one-dimensional array.
Yes.
And you think there are speeds at which the edge of a dot would release inhibition, and then you'd get some excitation before it. Right? I mean, if
yes.
Slowly enough, maybe, or
okay.
scale. So it's not clear to me that it should cancel.
Okay, you're right. So I think maybe if I can rephrase your question, I have the same question, and I think the same question applies to nearly any questions in question of this kind in neuroscience. I have always the same question. We have these units, these elements that have timescales attached to them. And nonetheless, we see a world where we don't have the impression that we're selecting very specific timescales. Yeah. Okay, so I think this is a very, very general problem. And somehow, things work well enough on some range of timescales. So part of the response is due to the fact that these excitation and inhibition are extended in time. Okay, so they're not very, very sharp. They extend. But that, in my mind, this is only part of the question, because if you think about, for example, very, very low light vision, you have photons coming in at a very, very low rate, so what you're getting is very discrete input, and nonetheless you see continuous work. Okay, so this is only a partial answer to your question, but I think it's a real question, but it goes much beyond what I'm saying. Okay, so no criticism. No? Okay, so the criticism you should have had is that you should have told me, well, I mean, why did you come up with this complicated thing here? All you've done here is to add dark in the receptive field. This is just an off cell, It's just a cell that measures how much light intensity there is and responds to more dark. Okay, so no need for that stuff. So we thought about this, and we applied some control. So what we did was an experiment where we have a movie. So this is time, this is space. So we have an expanding object. So we have a mimicking of an approaching motion. But at the same time, what we do is we make everything brighter gradually. Okay? And we do it in such a way that the light intensity remains constant throughout the experiment. Okay? And nonetheless, the cell responds. Okay? And the model obviously also responds. So if it were just a kind of dark cell, an off detector, then it wouldn't respond because nothing is changing in time here. But it does respond. Okay? And so the reason it does respond, what is really very essential here is two things. Is that I have these very small subunits. So they are good at detecting local motion and bad at detecting diffuse changes in time. And the other thing is that they sum. The sum over these subunits is nonlinear. If I had a linear system, I would have no hope of responding to something of which the sum is constant. Okay. Now, so really, what I think the moral of this story is that some cells can have very sophisticated composites receptive fields, And I think these are to be contrasted with the classical view, that's what you see in textbooks about the visual system, or at least the early visual system, where receptive fields are these stupid objects that are linear and have an excitatory center and an inhibitory surround. And for these objects, clearly, if you have a bar that moves through them, you will have a very strong response. Whereas in our case, we had no response whatsoever. So I can't resist to just pause here for a second and mention that the first guy, as far as I know, who thought about these center-surround receptive field was Ernst Mach, who was the same Mach who influenced Einstein for general relativity. And to me, it's completely mind-boggling how he could think of this, because he thought about these based on such illusions, which are now called the Mach bands. And these illusions, so you have a light area and a dark area, and what you should see is this kind of more white line here that really doesn't exist. So here the light intensity is monotonically decreasing, but what you see is something like this. You see a peak here. Okay. So one example is the white line you see on the horizon. Okay. So this dude, maybe there's some physical reason, maybe there's some light scattering, I think. So I'm cheating a bit, I suspect, but okay, let's say it's the Mach band. So this dude saw these things, and he thought, and he said, okay, then retinal cells must be center surround. Okay, now there was no physiology, there was nothing. This is very impressive, I think. So not only that, but he wrote that this is clearly the beginning of a caricaturing process of feature detection, of redundancy reduction, all these ideas that people even at Princeton work on. And so he predicted all of that, and he said clearly this is the teleological significance of this process is clear in itself. It is an analog of abstraction of the formation of concepts. So this is really very, very impressive. And I think this is what I was telling Howard. Looks a bit like Howard's office, Howard Stone, which is also full of books like this. So clearly he was well read. But I think also to do something like this, you need a lot of concentration, and he had to think very hard. And this is why I think that on many pictures you see his fists are really clasped, because he's focusing so hard. But it's okay because he got his stamp. So, so now a last comment I want to make about this circuit before I move on is that this is very cute. So this device that we talked about, this electrical synapse, it was so important to us between an unbipolar cell and this A2 amacrine cell. Actually, we knew about it already, or at least the retinal community knew about it, but knew about it in a completely different context, in the context of nighttime vision. So in the night, you're using your rods, you're not using your cones, and the way you do to pull a lot of information, to pull a lot of light on many rods, is that you use the cone system. You piggyback the rod system on the cone system, and you do this by using this synapse. But it goes in this direction, the information. So the way, so the rods will excite this bipolar cell. This bipolar cell excites this amacrine cell. This amacrine cell has a hole with this other bipolar cell. So this behaves like one big cell. Now this is an on cell for cones. So this will activate the same downstream circuit that you use during the day. Okay. So it uses this synapse as a kind of little network motif. But the information is flowing in this direction. And what I told you about our circuit is that it uses the same synapse, but in an inhibitory way. And the information is flowing in this direction. So that's very cute. That's an example of how nature is just packaging function madly in small circuits, okay, and using the same small circuit motif to to treat information very differently during the day and during the night. Okay, so sorry. So this I should mention just a small comment about context, which is that cells that detect approaching motion were known, but they were not known in retina. They were known in the brains of bird, of pigeon, and of insects. However, they're different cells. They behave kind of differently. Those cells are probably coding a collision, an impending collision. Whereas these cells we've been talking about code the onset of motion. And in a very complementary way, Eric here, Eric Chen in Michael's lab, has identified and is studying now a cell that is very complementary because it's a cell that can tell the onset not of approaching motion but of lateral motion. Not of lateral motion, not of approach motion, but of lateral motion. And so what it means is that this cell will respond much better much more strongly to a motion like this, where there's a bar that suddenly starts moving instead of a bar that is moving in a smooth motion. And what's kind of curious here is that the stimulus is the same on the right. The only difference is on the left. And so somehow this cell must use this part of the stimulus to suppress its own activity. And that's kind of paradoxical because this is also the stimulus that is animating the cell. So now with Michael and Eric, we're kind of discussing and thinking about what kinds of circuits and what kinds of models what kind of mechanisms can explain this. Another thing that I'd like to mention before moving on very quickly is that these circuits I've been talking about are kind of static circuits. They have some architecture and they're there to do a computation. But there are more sophisticated computation done by similar small circuits that are adaptive, so they change in time. And there's a set of examples that me and others have been thinking about, which are circuits that can adapt to specific features in the statistics of the input. And they do this with kind of more dynamical computation. So I won't talk about this, but if you want, we can chat some other day. So before moving on to larger networks, let me make some general points about this circuit issue. So the first thing is that it seems that the traditional view that we have for visual processing that textbooks give you, which is a view in which the retina makes a Xerox of the world by picking up pixels and then sends that Xerox, this linear photocopy, to the visual cortex, where you have in the primary visual cortex, you put the pixels together to construct bars, and you're sensitive to bars of some orientation. And then something happens, and in a feed-forward way, you get to the cells of Professor Gross that can distinguish the face of, say, a physicist versus more normal-looking people. And so this view, even though it's probably partly true, it's certainly very, very, very incomplete. So one obvious reason that everybody in this room has in mind is incomplete because of the massive feedback that you have at so many levels in this hierarchy. But another reason is that already in the retina, there are many, many sophisticated circuits. So they divide information in channels in a much more sophisticated way than this picture implies. And one can guess that this is actually very dramatic if you look at the diversity of cells in the retina. There may be 10, 15 types of bipolar cells, maybe 20, 30 types of amacrine cells, maybe 15, 20 types of ganglion cells. And so you can imagine with this how many different circuits you can make. And so the retina is kind of dividing the visual scene in many, many different channels and sending this to the brain, and somehow the brain has to deal with these. And when you see this, it's very hard to think that there are not similar computational devices also in the cortex. So the conclusions are that it's maybe meaningful, or at least it's certainly interesting, to identify these circuit modules. These circuit modules can carry very definite functions, can even carry more than one function. And there's some obvious question to ask is how many sensory features can you pick up? Coming back to the question of Professor Holmes, what kind of temporal and spatial scale do you have access to? How fine the statistics of the input can you adapt to or can you pick up? And how many circuits do you have? What are the channels in which you're breaking up your information? An obvious question also is how are these read out and recombined by downstream circuits? So of course I know none of these answers, but one thing maybe I should end with here is that all the examples I've seen of these circuits that detect various visual things are based on the same three principles. And these principles are the following. There is some generic form of excitation. Generic in the sense that it doesn't depend on the input that you want to select for. Maybe it has some spatial property, like in our case it was local in space. But it's not input specific. Then there is inhibition, and this inhibition kind of sculpts the response in an input or in a feature-specific way, if you want, by suppressing unwanted stimuli. And finally, you have a nonlinearity which is always key in these computations, either some thresholding or some nonlinear summation. And so this is very, very different from the way an engineer would do it. And if you wanted to pick up a specific feature as an engineer, you would first find an algorithm that finds it in the visual scene, and then you would increase the gain on the response to that feature maybe, or something like this. So this is doing it in a very different way. You're kind of exciting everything, and then you're suppressing what you don't want, and you're kind of filtering it out with a nonlinearity. Okay? And that seems to be a relatively common principle of all these circuits. Okay, so I'm doing all right on time. So this was more or less the first half of this talk, and I think I've spoken exactly 30 minutes. So now let's move on. I will now be a little bit more superficial, also because some of these questions are much more technical mathematically, and so I'll be forced to be a little bit more soft. Okay, so what I have in mind for when I speak about networks as opposed to circuits. Oh, first of all, are there any questions maybe on the first part? Okay, so what I have in mind when I talk about networks is possibly a larger number of neurons than in these small circuits, but mostly what I'm thinking about is some network where I don't have a good sense of the detailed architecture, like of the hardware architecture, if you want. Maybe all I know is some statistics of the connectivity, and I don't have a very clear idea of the cell type. So I don't know individually the cells how they behave in detail. Okay, and so the flavor of questions I'm going to ask, and the flavor of the thinking is completely different. I'm not going to look for mechanisms, specific computations, but rather it's kind of a vaguer work. So it's trying to come up with some bounds, some theoretical bounds, or some theoretical proposals on either how the dynamics is or how the coding is, and kind of propose these as ways to plan new experiments. So the main thing I want to discuss is some problems having to do with architecture and dynamics of cortical networks, and I'll be really thinking about the cortical area when I say cortical network. So again, I'm not going to focus on one very definite experiment like I did last time. I'm going to focus on a set of different data and extract from them a trend and not a specific result. And the trend I want to focus on here is the fact that neural activity propagates fast. Okay? So those of you who are familiar with the work of Thorpe and others, you know that the information, the work on, I'm sure you know this much better than I do, but the work on, say, recognition of animals in pictures, even more strikingly faces, you can extract from these data that information takes maybe less than 100 milliseconds to go from the back of the cortex to the frontal area. So it's very fast. What I want to focus on more here is lateral propagation in a given area, like V1 or S1. And the rule of thumb is that it also propagates very fast, and roughly it propagates over 1 centimeter in a few tens of milliseconds. Okay? So the kind of data I have in mind is this kind of front propagation. I'll come back to this and I'll tell you more about it. I don't want to explain it now. This is in rat, and it's a bit slower than this, but in cat and primate it's more like these numbers. Now, this is fairly large, okay? In one of these you have 10,000 neurons. This is fairly large, so you can't propagate through this whole thing with one synapse. So presumably you have to go through many synapses, okay? So somehow there must be short polysynaptic paths. By short I mean with few synapses that allows you to propagate very far, okay? And so what I want to ask is, how do these paths emerge? Do they have to be hardwired? Do they have to be designed like an engineer would design them? And the other question I want to ask is, what do they imply for dynamics? So what I'm going to do is just to write down the simplest model I can think of, and then try to think about these paths. Okay? So the simplest model is this. I'll just put down a number of I'll put down a number of neurons with some surface density. I'm imagining again that I'm thinking about the cortical area. And I'll assume that locally these neurons are well connected, and they're more sparsely connected more remotely. That is true because, of course, there are fewer long axons, there are fewer long connections. So specifically, the way I'll write this down mathematically is I'll say the probability of connection is a constant nearby, say in a ball of 100 microns, and this constant is about 1/10 in the cortex. And beyond that, I'll say that the connection probability drops like a power law with some power sigma, like this, okay? And this is, one can argue whether this is a good description. There are some data that suggest that this is steeper, this is maybe more exponential, but there are a number of data that kind of suggest that such a fit is not too bad to describe the data, and with these numbers. So the dimensionality of my system is 2. If I think of the cortex as a flat sheet, if I insist on including a lot of circumvolution, maybe it's 2.5 or something, and sigma is roughly 3. Okay? Now, this is not, the physicist's reaction to this is to think about asymptotics. This is not my point here. My point here is I just want a good fit over some range. Okay?
Does it matter how it must matter how you put down those points.
Well, I put them just randomly, but they're very dense. If they was very sparse, of course there would be fluctuations that would matter, but it's so dense.
Just a Poisson.
Yeah. It's completely, yeah, exactly. But it's so dense that those fluctuations kind of average out. Okay. So this is the main message, maybe, of this piece on architecture. So suppose, remember, I said we're propagating very far, so I have to have a path that goes very far. So suppose I want to go from this seed neuron here to this target neuron there. Okay? Now, it's very unlikely that there will be one connection between the two. It's super unlikely. Okay? However, this neuron is connected to many local neighbors. So what is likely is that one of these neighbors will be connected to the target. Okay, that's much more likely. So how likely is it? Well, I will find such a path if the number of local neighbors here times the probability of having this long connection is of order 1. Then it will be likely that such a bisynaptic path exists, right? So if I call zeta the number of local neighbors, and I just plug in the formula I had before for the probability, I can estimate the scale of bisynaptic paths, and this scale would just be this size R0, this scale in my model, times some number which boosts it, and it will boost it as a function of the number of local neighbors. Okay? Now, you can very well imagine that if I now were thinking about not a bisynaptic but a trisynaptic path, the advantageous thing to do would be to consider the local neighbors of my seed neurons, the local neighbors of my target neurons, and then to connect the two. Okay? And if I think about a path with more synapses, I can just nest this thing. So now I have local neighbors here that I can reach with 3 synapses, local neighbor there, and a long bond between the two, and so on. And if I can just keep doing this, and at each stage I could do a little calculation, and I could do the recursion, and what I would end up at the end is such a formula. So I don't want you to focus on the details of this formula. The details maybe are not so important. What is important is the rough behavior. So what we end up finding is that if I want to go from one spot to a distance r with a polysynaptic path, the number of synapses I need grows very, very slowly with the distance. It grows like the log to some power. I think it was the Russian physicist Lev Landau who said that a chicken is not a bird and a logarithm is not a function. So what he meant by this was that the logarithm is essentially a constant. So it doesn't matter what r you put in, okay? And so that's what you see in this plot, is that the number of synapses as a function of r grows very, very, very slowly. Another way to say it is by allowing one more synapse, you can go much, much, much farther, okay, in an area. The other thing is that it's because of this logarithmic behavior, it's very robust to the parameters. So if I multiply, say, by 10 my local connections, I just gain maybe 1 or 2 synapses. Okay, not much more. So this is kind of instructive as just of how to do research, which is that if we had not thought about this and we had thought, oh no, like pyramidal cells of such type. We don't know if they have 500 local neighbors or 800, so we can certainly not write down a model. Then we will never know that it doesn't matter at all. Because you multiply something here by a factor of 2, nothing changes. Okay? So again, here what I want to say is not a detailed result. I don't want quantity. I don't want to tell you, oh, if you want to go from here to there in the rat, you need 3 synapses. That's not the kind of result I'm aiming at. What I want to say is that if I have a simple model like that with local connections that are pretty good, like a 10th connectivity, and long-range connections that are very sparse, but not so sparse that they're, say, exponentially improbable, then these very efficient paths emerge, just statistically. You don't have to design them. Okay. So I have simplified things quite a lot in this discussion. Of course, things are much more complicated. One can prove somewhat rigorously that these paths are the ones that optimize things. Here I've just derived for you a scale, but you can actually derive the statistics of these paths, the probability of these paths, and you get corrections to this scale, but none of these are very meaningful qualitatively. What is... Yes?
So I don't want to make you get too specific.
Yes.
But I'm thinking of visual cortex, and I thought that the P of R was patchy at the root. So instead of dropping off, it has a bump, and then a long distance another bump, and a long distance another bump.
You're right.
Wouldn't that completely...
Okay. So I think... Okay. So let me backtrack and say... Okay, what does design mean? Suppose you want to create a network and you want to design it. The naive thinking of design is I take my pencil and I draw some network. Okay, so now I've determined everything. There's no statistical knowledge. It's all determined. Okay, if I want to go from the other end, the least amount of design is a completely random network where I have 0 parameters or 1 parameter, let's say the connection. Okay. Then I want to design it a bit more. I'll add 1 more parameter, this fall off, for example. Then I can add 1 more parameter, which is this clumpiness. Okay? So I'm saying if you want, you can think of this as a mean field version of what you're saying. Okay? Like if you average over the clumps, what you would get is a distribution like this. But I agree with you that what one should do, and actually I think I mentioned it in a slide later on, is that for a finer model you would want to take both these anatomical fluctuations and some dynamical fluctuations into account. So you want to take into account these clumpiness, these arborizations, and so on. But you could think of this as an average version of what you're thinking about.
You can't average the network before you calculate what computations you do.
It depends. That's right, but it depends what quantities you're looking at. So it depends because
Taking the average transistor.
True, but it depends. So sure. But it depends what kind of computations you want to do. So maybe when I come to the dynamics, I can. Okay, maybe. Okay, let me get to the dynamics and then we come back to this question, if you don't mind. But it all depends on the quantity. If you want to calculate a mean field like quantity, it's okay. If you want to calculate a quantity that will self-average, it's okay, right? Depends on which quantity, I think.
If I want to calculate how much heat the CPU uses, then yeah, the average transistor will be fine.
Yeah. Okay, but
But you still can't do it calculating on the average circuit.
No, I think so. Here, let me comment on it now. So what I'll get to is a problem in which I want to sum over paths. Okay. So if I sum over paths, and in the sum over paths, this implies the kind of averaging I'm just talking about, I think it will be okay. And I'm not sure it's okay in this case, but I'm not sure it's not okay. Okay, okay, so maybe a more meaningful kind of mathematical detail I should mention for people who are interested in it, maybe you will be interested in it, is that this regime I've been telling you about is a very non-trivial regime. It's only true for this fall-off power, sigma, between d and 2d. Okay? If you have a fall-off that is too shallow, too small, then the network just behaves like a random graph. And roughly everything is connected to everything. So you can go from any point to any other point by using 1 or 2 synapses. So you have very efficient paths, but you lose locality. You lose the notion of locality. On the other hand, if you have here a very steep decay of the probability, of the connection probability, now you keep locality, but more or less your network will behave like just a lattice, where if you want to go to a distance r, you have to go through a number of synapses that scales like r. So you keep locality here, but you lose efficiency. So this intermediary regime is kind of very non-trivial and very nice because you keep some idea of locality and you have very efficient paths. Okay? And kind of interesting things happen here and so on, but they're not so relevant for this discussion. So what about implications for dynamics? So remember we said that the rule of thumb, I want propagation by 1 centimeter in 100 milliseconds. Okay, and I think conceptually a problem that is more important is that I want propagation that is very sensitive. Okay, if I deflect my whisker a little bit, I want to be able that my cortex can pick up that information. But if I deflect my whisker in the rats by twice as much, I don't want the whole cortex to go into epilepsy. So I have to have a system that is so sensitive, but at the same time very, very stable to increasing the input. So that's a kind of a non-trivial problem. I want a large gain, but a stable system. So, sorry. So this is what I have in mind. These are the kind of experiments I have in mind. You take a rat, and you deflect one whisker by some angle. And what you see is that the barrel that corresponds to this whisker lights up. And then, within 20 milliseconds, you see some propagation. Within 50 milliseconds, you've more or less covered the whole barrel cortex. Okay? So to fix the ideas, theoretically, what I'll be thinking about is some seed activity. And I want to ask how this seed activity creates an advancing front. What is the behavior of this front? How fast does it go? How far can it go? Is it stable or not? So I will not have any time to go through any kind of detailed discussion of this. Let me just try to give you some intuition and some preliminary results. So if I want to know the behavior of the front, it's maybe a good idea to focus on the neuron that is sitting on this front. Okay. And so what happens on the front? On the front, this neuron, this target neuron, if you want, receives a lot of local excitation. And as we said before, having these kinds of paths boosts the number of paths that you have access to. So there will be a lot of excitation that arrives on this neuron from local neighbors. Okay. Now, if you just had that, as you're looking at longer and longer distance, the number of paths becomes so large that you would have a runaway activity. You would go into epilepsy, epileptiform propagation. So we have to invoke some amount of inhibition to stabilize this behavior. In fact, we have to invoke so much inhibition that beyond some scale, these local neighbors will not be enough to propagate the front, and we will have to rely on long-range bonds. But these long-range bonds become rarer and rarer as as my scale becomes larger and larger. So if you take this intuition and you kind of massage it mathematically, this is the kind of results you get to. So first, we find that there is no propagation if you don't stimulate your system enough. If you just hit it a little bit, nothing will propagate. If now you go above threshold, we find the propagation in the right order of magnitude, okay, which is, I don't know how important it is, but it's at least satisfactory that we don't get absurd velocities. Maybe the more interesting thing in my mind is that the stability that we kind of got out of inhibition will have as an effect that the span of the propagation is relatively insensitive to the input. If I double my input, the span will change just a little bit. However, the speed will change a lot. So that's kind of a strange system where by increasing my input, I make the thing go much faster, but it stops more or less at the same point. Okay, that has to do with the nature of these paths. And this is kind of what you see in data. I don't want to make too strong of a claim. I just want to say it's kind of nice. It's kind of acceptable. So if you deflect a whisker by 0.6 degrees, you see some activity in the corresponding barrel, but no propagation. If you deflect the whisker by 2 degrees, you see propagation, and so do you if you deflect it by 6 degrees. But if you look, say, here, after about 20 milliseconds, if you look here, you have propagated much farther with a bigger input than with a smaller input. So it kind of agrees with this. But all this is very preliminary, yes?
Just to play devil's advocate, there's this funny result in, if you look in anesthetized mammalian visual cortex cat or monkey, if you're just flashing a Gabor at varying contrast, you find actually at the higher contrast stimulus, you're jazzing the system more, actually gives you, reduces the spatial extent to, say, traveling field potential waves. And if you sort of look at a spike-triggered average in spontaneous activity, and you think about it as sort of a zero contrast condition, that's where you get the broadest network activation for a given spike.
Right.
So how do you sort of reconcile that with...
Okay, so I'll come to that. So I'm not familiar with the first result of the two that you mentioned.
So the idea is you crank up the contrast, you get more and more localized activation.
Okay, so I haven't seen this result. I'm sure it's correct, but I haven't seen it. There is very analogous result to this one done in monkey with optical imaging by Greenwald and company, and it's kind of analogous to this. I don't know if they've looked at contrast dependence. That I can't remember. So I don't have a good answer, but I'll say a word about it just in 1 second if you, maybe next transparency or something. So I just still want to say that there is one problem here, is that this threshold we find is very large. So basically we find that you really have to hit the system you kind of really have to light up the whole barrel in some sense with your whisker before things propagate. So it's not that sensitive. And with the crude model we're looking at, there's no way out of this. And I think this relates to what you're saying, maybe. So let me, I have a guess, but that's all I have. Okay, so let me just conclude this. So the main conclusions are that if we want these efficient paths with few synapses, we don't have to design them. They can emerge statistically, and they have a very non-trivial, very surprising implication for dynamics. Now, of course, there are many more open questions than there are conclusions to this piece of work. Of course, the most interesting maybe is, can we go and look at data? I don't think viral tracing, for example, now is good enough, but maybe at some point we can look at these paths and see if they're overrepresented, or if we can find their statistics and so on. Another thing, theoretically, of course, one would like to include the kind of stuff you're saying. So that's what I mean by axon structure. These arborizations, these clumpiness, the stuff you're mentioning, also finer neuron dynamics. And maybe relating to your question, and the reason why I think maybe it relates to this threshold business, is that here, what I've looked at, and that maybe has to do with your point also, is I've really looked at some kind of mean field model. So I've looked at propagation of mean activity, excitation minus inhibition, if you want. Okay, and that's why I think that maybe it's okay to average over the network because of this quantity I'm calculating. But maybe what is really happening is that really inhibition suppresses everything, and we have to think about propagation of a fluctuation and not of a mean activity. And that kind of agrees with what you were saying, because then no contrast, you would only see the fluctuations. And with high contrast, you're kind of squishing the fluctuations down. You're just increasing inhibition and you're just suppressing everything. Okay? And so my guess is that what I should do is to think about an analogous problem where what is propagating is a fluctuation. Okay. Yes. Yes.
So, I mean, you said that the span should be insensitive to the intensity, but it just doesn't look like that's the case.
That is true. So in this data, it doesn't look like that's true. Of course, it's not very clear what the span is here, and so there's no larger thing. But I agree that this is less clear. I didn't mean that what I meant really by saying this is that the dependence is much stronger for the speed than the span, and maybe a more precise statement here would be to say that initially for small stimulus there's a regime where the span depends on the stimulus, and for very large stimulus you're kind of plateau. Okay, and so maybe here you're still in this small stimulus regime. It's not a very precise way of saying. But I agree. I mean, I haven't. Maybe it agrees, maybe it doesn't. Ultimately, I'm not sure. I would have to see more data and compare more precisely with that. Okay, so in the should I stop here, or should I just show you briefly something on single cells? You tell me. Yeah. Yes.
Say a word or two more about the model that produced these results. I mean,
Yes. So
it's a firing rate model.
yes. So I was hesitating whether I should say it, but I thought it would carry us too far. So maybe let me say one sentence about it, and then if you want, we can discuss. I would love to discuss it more in detail. So I'd be very happy to do that. So this is the crudest possible model that we could think of. So this is how it goes. You have a neuron that fires, projects on another neuron. When this neuron gets a spike, it has a probability f of firing, okay? And then this will project to another neuron, then it's probability f squared, everything is linear, okay? And so then there's a second, if you have a second path here, you also have f here, and if you get there, you have two f's because you have two paths. So basically, the firing probability of this neuron is f to the power of the length of the path times the number of the paths times the number of neurons that were excited initially. Okay, and then you have to include inhibition and so on. So as I said, this is a very, very crude model. You could imagine 1 million refinements to this. Okay, so I think maybe because it's so crude, it saves me for your question. Okay. So yes. Okay, so should I stop? Should I not stop? Okay, I will skip this. This is work on coding that I've done with Michael, and I think you've heard about it from Michael. We kind of showed that if you have correlations in your neuron and diversity, physiological diversity, then you get wonderful things. Okay, so let me spend just 5 minutes or something on this. So when people like me who have a physicist background go and look at these large networks, it's a little bit unhealthy, the mode we get in, because we start thinking about neurons in these very crude terms. And I think it's very healthy, and it's also very pleasureful, actually, for me. It has a lot of pleasure to go back and realize how complicated individual neurons are. And here, this is just a study case on photoreceptors. So as you know, photoreceptors are your interface with the visual world. So they capture light and produce some output that is transmitted to your visual system. And what is in them? It's a page from our lectures with Michael, from our course with Michael, which describes how this molecular network works inside the photoreceptor to transform light into some current in the outer segment. So I don't want you, I'm not going to discuss this. I just want you to see how complicated it is. There's this feed-forward cascade, phototransduction cascade, and then there are 1 million feedback regulation loops, some of them mediated by calcium, others some clever biochemical tricks, and there are really tens, maybe, regulation loops and mechanisms. They're not all well known. The parameters are not measured, and more fundamentally, if you model this, you get many, many coupled nonlinear equations. And so there is no hope that by looking at these equations, you can tell how the photoreceptor behaves. So what we did, we said we're going to go from the opposite extreme and just guess the simplest equation we can think of that will describe a photoreceptor. And this was the simplest equation. So if you have the response of the photoreceptor is some low-pass filtered version of y, and y is simply some linear filtering of the light input. Okay, so that's the simplest thing you can write. Of course, this will not satisfy the problem because there's no gain control, there's no adaptation. And one thing we know is that at higher light level, photoreceptors will decrease their gain and become faster. So what is the simplest thing we can add to this equation? I think the simplest thing is to add this one term to the relaxation term, this multiplicative factor to the relaxation term, that is itself, again, some linear filtering of the light input, maybe slightly delayed compared to the y term. So it keeps some memory of the input. Okay? And this will make the gain lower and make the photoreceptor faster. So this is kind of the... Biologists love these things. They're very confusing, I think. So this is a picture of this model. So you have two channels. You have one channel which filters the noise, filters the light input, low pass filters it again, and then another channel with this Z term that kind of modulates the low pass filtering. Okay, so if you compare this model, this is a model, this is data for responses to flashes, you see that there's very good agreement. Of course, here we can take advantage of 40 years of research on the photoreceptors. And we found when we put very intense light flashes, we found this kind of non-monotonic response, and we thought, okay, the model maybe is really wrong at these high light intensities. Then we went back and looked at the literature, and we find them in certain cone when you really blast them with very high light flashes. So that was kind of nice. Also, if now you don't show flashes of different sizes, but the same flash on top of light backgrounds, you see that the model agrees very well with data. And what I want to emphasize here is what happens. So first of all, the response becomes faster. The peak moves to the left. And the other thing that happens is that at low light levels, in this dim light case, you just have an integrator response. And at high light level, you have more of a biphasic response, so more of a differentiator response. And this is also what you see in the model. What you don't see on this diagram is that the gain goes down because all these curves have been renormalized to have the same maximum. But if you measure the sensitivity of a cone and you compare it to data, you see this nice agreement over like many, many decades. Okay. Yes. Great.
So the model relies on two kernels.
Yes.
KZ and KY.
Yes.
And you're choosing them in some way. You're inferring
so I'm choosing. I don't like fitting. So I take the simple. So I take just.
You invent something that.
I take what everybody uses, which is this. Let's take, say, t to the power n divided by tau to the power n plus 1 n factorial, just to normalize it. E minus t over tau. So it's just essentially, these all kernels are this. So you have some rise with power n, grows like t to the n initially, and it decays exponentially. Okay, so this is a comparison, but then we said we would compare it to more kind of less stereotypical stimuli, not spots or flashes or steps, but we'll compare it in the case of flicker. So in the lab of Marcus Meister, they measured the response of salamander photoreceptors to flickering light. That's what they measured. And this is a comparison. So the red is our model, the black is the data, and as you can see, it agrees kind of nicely. And what is the blue? The blue is the response, is the output of a very simple model that everybody uses. And I thought it was a nice benchmark to compare our model. And it's, again, this linear-nonlinear model where you kind of find a filter and then convolve it with a nonlinearity. So here you do have to fit a whole function, actually, which is not the case in our model. In our model, you just fit 2-3 parameters. Actually, our model is more impressive than this. You can replicate families of curves. I'm skipping a lot of things here. And you see that our model kind of corrects for systematic mistakes that the LN model does. The reason it does these mistakes is that it's not really adapting to anything. In our model, we have this memory kernel that keeps memory of what's going on and adapts to it. So this is kind of unimpressive, this difference, these corrections. You may think, who cares? The reason it's unimpressive, I see David Tank smiling, so he probably thought, who cares? But the reason it's unimpressive is because this contrast is very small. The standard deviation is maybe 30% of the mean, okay? But in natural vision, when you're walking in the woods on a sunny day, the fluctuations are huge, because at some point you're looking at the sky, next you're looking down, at some shadow, and so the fluctuations are huge. Nonetheless, and this is a model output, the photoreceptor adapts, is continually adapting so as to keep a dynamic range of a factor 10 or 100. Okay? And so we thought, how can we see really how much the photoreceptor is adapting as it's dynamically, how does it adapt as it evolves? And we thought, well, there's a simple thing we can do. We just superimpose little flashes here on top of this and just calculate the flash response of the photoreceptor. And to our surprise, this is what we found. Forget about the thick lines. Just look at the thin lines. So these are responses to flashes at different moments in this natural vision. Okay? And they change by a factor 10 or 20. So you're walking in the woods, and the time scale, the time scale of adaptation is about 300 milliseconds, which, by the way, is close to the time scale of saccades. So you're walking in the woods, and your eyes are moving around, you're saccading. Every saccade that you do, you spend 200, 300 milliseconds, you're adapting to fluctuations, and you're adapting in your photoreceptors, the very first set of the visual system, by a factor of 20, every 30 milliseconds, or 5 or 10. Okay, so this kind of rapid adaptation was not something I think that people fully realized before, and they were using these flickering inputs thinking this gets rid of any adaptation, but that's not true. The photoreceptor is constantly adapting. So there's rapid adaptation, and you could think about it as some example of active sensation, because by moving your head, you're making your photoreceptors adapt more or less. Actually, it's more detailed than this. You can even show that your photoreceptor, in some sense, is adapting to variations. The adaptation depends on the temporal correlations. I mean, we could go more into it. I think maybe the more conceptual conclusions, the more meta conclusions, if you want, are the fact that you can contrast the view that a biochemical model gives you and the view that a phenomenological model gives you. And these are very complementary views. And traditionally, people would talk about these cells as having a response and then adapting on top of it. But maybe it's more natural to think of these cells as coming with some time scales and some nonlinearity, and that captures both the response and the adaptation that occurs on the same time scale. The other thing which is kind of amusing to me, which was amusing, is that when you look at this phenomenological model, you realize that a lot of the adaptation is done in a feed-forward way, not by these loops, say the calcium-mediated loops. And initially, like 30 years ago, when people were starting to study adaptation in photoreceptors, all they were thinking about were these loops. And it took them a long time. So it's only in the late 90s, early 2000s that the biochemical community realized that a lot of stuff is happening in just the feed-forward cascade. So these phenomenological models kind of allow you to have these conceptual shortcuts. The other thing which was nice for me is that it led to much more work. So we kind of generalized this model to have much more elaborate models for adaptation with more elaborate timescales and so on. Now we're thinking about applying these models to actually circuit models, so having circuits of these cells and so on. So let me conclude, but just before concluding, I just want to mention one thing. There's a set of other work that I won't have time to talk about today. I just wanted to mention this because some people here may be interested in these problems and I'd love to chat about them. So this is some idea we had about how postsynaptic domains can form and be stable just from biophysics. And what we found, to our surprise, is that the diffusion of receptors, which you would think should destabilize molecular domains, the diffusion of receptors on the membrane, which one would naively think should destabilize these molecular domains, we find it actually they stabilize them and allow some form of plasticities that we speculated upon. So we've published something on this recently, and now I'm thinking about a completely different model that explains the same thing. And I would love to chat about it just to mature my own ideas. So this is the end, but let me maybe just before really finishing, I will mention another reason why I gave this title in reference to Leopardi's play, 6 Characters in Search of an Author. And the reason is the following, is that in every... not Leopardi, Pirandello. So in each of these problems I've mentioned, you could ask me, how does the readout work? Okay, you could ask me, how is this information collected and analyzed by downstream circuits? And as far as I can tell, there is no good answers that theorists have to this question, because what we always tacitly assume is that there is some kind of super neuron, some kind of grandmother cell that is watching everything and making a decision, or making some end of the line computation, some kind of brainonculus that is doing it. And we keep saying that the brain is not a set of grandmother cells and so on, but I don't know what the alternative is really, or what I have never seen an interesting alternative to it. And so it's a little bit like this play where everybody is reading out everybody else. So the actors play the characters, the character correct the actors, the director is himself an actor, and the play is actors practicing a play of the same Pirandello and so on. And there also there is a super neuron, and these are the spectators who are seeing the play, and they know what is going on. They know it's just a play and they're just actors. So there is the same problem, and it's not solved. Only in theater that's the way it should be. Thank you.
Questions? So if I go back to the network problem, where you show the P of R, suppose that as an optimization problem without making assumptions about connections, but had that as design variables. And let's say one sort of constraints. And you pose the right objective. What would you get for distribution?
But it would depend really on your constraints, I guess.
Of course. I'm just wondering, have you thought about it?
I mean, the thing is, you would have to come up with...
The important thing is coming up with the objective function.
The problem is you would have to come up with a very non-trivial constraint function, I think. Because any trivial thing that you would say, you would get probably something like a random network. No.
Depends on
like suppose, yeah, I mean, so I think maybe the way to do it is to what kind of constraint function you would want that would keep locality and allow you to have very short paths, something like this. So maybe that's